The first speaker, I don't ever have a keynote, but I was thinking, who should I have first? And 5 years ago, I had a presentation by Bill Haseltine, and I still remember that speech. It was a fantastic speech, and he never mentioned his very successful company. It obeyed all the rules of my conferences, of not doing a sales piece, and of looking at what's next in the future. And I remember that speech very well. So when I was thinking of this conference, I thought I'd have him first. It turns out very lucky that I had him first, because right after his speech, he has to run off and fly to England anyway, which just came up. So if I hadn't gotten him first, I wouldn't have had him at all. Anyway, Bill Haseltine. Just for the moment, that's fine.
Can we have the lights up a little bit, if that's possible, so I can see who's out there?
The house lights go up.
What?
That's the most the house
Okay, well, I
We
can...
can turn these down, though, a little bit.
All right, and I'll see you in a minute. I think the first thing I'd like to comment is the bittersweet nature of this occasion. All of us who have had the privilege of attending the TED Med meetings or the TED meetings know what they mean to the community, know what a contribution they have been to our thinking in our own areas, know what they've been as a contribution, especially to lateral thinking, where we have the opportunity to meet with people that we don't ordinarily meet with, to be inspired by their visions and to carry on to do things we might not otherwise have done. And so I'd like to begin by recognizing our fearless leader, this time without a scarf, and I think we all should give him a round of applause and thanks, because this is his last TED meeting. Now, we need no more vivid reminder of what the human body can do when it's young and healthy, when it's older, than what we just saw. I don't know how many people in the audience could get up and do that, no matter what age we are. I know I don't think I could. But the goal that we all share is to allow us to do that at whatever age should we wish. To use our physical, our mental faculties to their full capacity, no matter what age we may be. And that is a goal that unites us as a group today and unites us as a profession. I thought I'd use this opportunity to look back on some of the highlights of the last 25 years where we've been successful, and I certainly can't mention all of those, and look forward to the next 25 years and to see where we may be successful and where some of the problems and roadblocks may lie that must be overcome. The first thing to say is the last 25 years has been an amazing time for medicine. It's been a time in which entirely new vistas have opened for the treatment, the care of those who are ill, and the maintenance in good health of those who would otherwise be sick. We have seen the advent of at least 2 and probably 3 new treatment modalities. 25 years ago, although we were treating a very few people with human proteins as drugs, it wasn't a systematic part of our medicine. It was limited almost exclusively to plasma from the blood, certain blood factors, and insulin that at that time was not human. The discovery of DNA, the discovery of the universality of its code, the discoveries that led to our ability to remove one gene from a human or other species and implant it functionally, purify it. That pioneering work has led to a new generation of medicines. Today it comprises about 15% of all medicines that we use. Those medicines make a real difference to patients' lives. And if we look at the future of human proteins as medicine, we see it is very bright. Because now we have the complete or near complete catalog of human proteins before us. And our task is not so much finding those proteins, which it certainly took a great deal of effort in the past, but to finding how each of those proteins fits into normal human biology and how they can be used to improve that biology. That's a definable task. It's a task that fits well within our medical expertise, our expertise in understanding cell biology, our expertise in disease pathogenesis. And we can see systematic and continued application of those technologies to ever-increasing areas of medicine, where we don't just change for a short time the chemicals in our body, but we restructure cells, we regrow cells in response to specific proteins that we've introduced, replacing those that are missing, adding those that need to be added. A second branch of medicine, more recent than the first, but nonetheless very impressive and growing extremely rapidly, are the use of our human antibodies as medicine. Now, once again, that isn't entirely new. Passive immunization was known for some time. We used sera from horses and other animals to help protect us in times of acute need as a consequence of infectious disease. But the ability to isolate a single human gene, to make an antibody, to manufacture it in bulk, and to do so with relative ease outside of a person, now even outside of an animal, has opened a very powerful branch of medicine. Because not only can we use those antibodies for the purposes for which nature has primarily designed them, to bind to foreign substances and to remove them, to flag them for destruction, to physically block them. That's what they do in infectious disease. But we can also use them to mimic natural substances. If you look at the way natural proteins interact with one another, they do it in a way that is very different from the way a chemical interacts with a protein. A chemical may have a 50 square angstrom, 100 square angstrom, sometimes even 200 square angstrom surface of interactions. Proteins, 1,000, 2,000 square angstroms of surface. It is those broad surface, weakly interacting surfaces which give much of the specificity to our living structures. We now know that we can mimic those structures with antibodies. That we can take what was a protein, create in a test tube a surface which looks like part of the important interacting surface, and use the antibody. That has several advantages. We can pick the parts we want, ignoring the parts we don't. We can create antibody drugs which will last in our bloodstream for many weeks, certainly several weeks, up to 40, 50 days in some cases. And so we have a two-armed aspect to these antibody drugs. On the one, they may be inhibitors, allowing us to remove or alter the concentration of natural substances. And on the other, they can be used to mimic and extend and refine the range of these natural substances, working for the most part as these substances do themselves. Another aspect of modern medicine that you will certainly hear a lot about today is new techniques in surgery. Those collections of methods that together amount to a reopening of the human body, allowing us to do deep within our body with micromanipulation, with endoscopic views, with tiny probes, that which we could only with the finest surgery do at the surface of our bodies. We could begin to imagine that 25 years ago. Today, that is a reality. And it is a growing reality for every part of our body, including the deepest recesses of our brain. Anybody who has had a friend, a relative, or has undergone surgery, him or herself today, knows what those advances mean and can foresee what many of those advances may be. We are on a very rapidly growing part of that growth curve. And any summary of our past successes over the past 25 years would not be complete without a description of what has happened to diagnostic imaging. As we can manipulate what has gone on in our body, so too can we perceive what is going on, and increasingly what is going on in real time. Whether we're doing real-time MRIs, real-time PET scanning, going to shorter times, going to ever more sophisticated methods, we can now peer into our bodies to diagnose, to observe, to correlate with disease symptom, and even to correlate with mental state. For the first time, we are beginning to build up detailed maps, functional maps, of how our brain works as it is working. And once again, we are on the early phase of a very rapidly growing curve in these technologies. All of this we have witnessed in our professional lives. What are we going to look forward to? What can be better than what we've seen? Always when you're at the bottom of a mountain, it looks higher than when you're at the top. So if my next comments come with some caveats, bear that in mind. I believe the next mountains we can climb will look shorter once we've climbed them. But some of them don't look very short right now. Let me take one area of medicine that is very exciting in its potential, and that is the use of nucleic acids as drugs. And that is a very broad field. One form of it is called gene therapy. Another form of it is called antisense therapy. Yet another form, and a very exciting new form, are called silencing or interfering RNAs. We have seen some partial successes. But for whatever successes we've seen today, they certainly have fallen far short of our hopes that we would be able to take our newfound knowledge of genes, what they do, and the ability to insert that, to use that, to manipulate at the genetic level for our own medical benefit. What are the key problems? The key problems that we perceive today with gene therapy are getting the right gene at the right place at the right time that is regulated normally. Now, we have certainly learned a great deal about gene regulation, but we haven't learned enough. We don't know how to get a right gene in the right place to make the right amount of stuff under most circumstances. There are very promising leads. I believe over the next 20 years, we will make those critical advances. There may be people here who are working on those problems, putting the genes into the cells with high efficiency, getting them to express under the exquisitely fine control necessary. The problems we have seen, and I'll refer to a specific problem with the treatment of immunodeficient children. The success of that treatment, as it happens, was very closely tied to its failure. It is very likely, at least in my opinion, the reason that that gene worked, was able to produce enough enzyme to overcome this inherited deficiency, was the very reason that ultimately failed. Because in order to get that cell to produce enough of that enzyme, that cell had to outgrow its neighbors, because we weren't able to get the genes in efficiently enough. So its success and its failure were coupled. We have to learn to overcome that. But I think we will. So too with antisense and maybe its more promising cousin. Maybe it's more promising because it's recently appeared. Maybe it is actually more promising because it more closely mimics nature, are interfering RNAs. Certainly as a research tool, it is enormously powerful. The ability to take a small snippet of RNA, place it on a cell, and turn a gene on and off at will. Were that we could do that for our own bodies. There we know what some of the problems will be. How to get those genes or those RNA snippets in the right place at the right time. How to make them as specific as we know they must be to do only what we want and not something else. But I would predict that with gene therapy, these RNA technologies will too, over the next 25 years, find a very important place in our pharmacopoeia. The next area of interest is the use of our cells. We can use our proteins. We can use our antibodies. Can we use our cells as medicine? Well, to some extent, the answer is yes, we know we can. We do autologous transplants. We take skin from one place, use it in another. We take tissue from one place and use it in another, blood vessels and muscle and other tissues. We know under some circumstances we can do that. We do that even from person to person. When we take a bone marrow transplant and take a stem cell from one person and use it in another. How broad will this application be? How useful will our newfound ability to create embryonic tissues from individuals or from a representative sample of our total species be? Those who are involved in this field are extremely hopeful, as am I. I believe that our experience with bone marrow transplants can eventually be repeated with almost every cell and every cell type in our body. But the distance between what we know today and what we must know in order to do that is enormous. Those quick routes through those northwest passages of modern medical research have run into solid walls. We, again, as in the case with our DNA and gene therapy, don't know enough about how cells are regulated. Don't know enough about how they speak to their neighbors. Don't know enough how to grow them in our cultures so that they will integrate properly when they are placed in the body. We will have successes. Some would argue we've had some successes over the past several years. But it is this area which is one of extreme promise. And it is the area where I counsel all young people to move. Not all, but those who are inclined to do modern developmental biology and cell biology. Because that is the next great frontier. We have in our hands, in my judgment, the fundamental tools that we need to make those advances. That is, we have the ability to create these cells. We have the ability to observe their changes. We have the ability to manipulate them in experimental models. And we will have the ability to move from where we are in our knowledge to where we must be over the next 10 to 25 years to integrate our cells as an important part of medicine. So too with material sciences. When we look at the path of material sciences, we've seen successes, starting from very early successes of using splints to help heal bone, to now using internal splints. We see an explosion of prosthetic devices used externally and internally. How rapidly will we be able to extrapolate our success in the material sciences to the integration of those materials into our body? Certainly it has happened. It's happened at a rather crude level. There are a number of materials we know are compatible with our body. There are a number of materials we can place in contact with cells and get them to do some of the things that they should do. And there is certainly the promise, and I think the very tangible promise, of being able to recreate most, if not eventually all, with perhaps the exception of the brain, our organs in culture for reimplantation out of our own cells. But it is going to take many, many years of hard work on each and every specific system. And it will take teams of people who understand the material, understand the cells, understand the medical problems to bring this into reality. Fortunately, if we look around our universities, we're beginning to see such teams assemble. They form nano, bio, whatever you want to call it. They are being formed as we speak. And fortunately, we have some visionary government leaders who are putting some funds into these groups. They will pay off, but there are as many or more problems to solve than have been solved. These are visions which can be realized. And I will finally close with one other area, the interface of our bodies with the electrical world. You heard that you're going to hear some outputs from those dancers and analyze it. Well, we've realized that human beings are part electrical for a long time, for well over 100 years. We've realized the positive and, I might say, negative aspects of interaction of the human body with electricity or with microcircuitry at this point. But we are just beginning a very interesting journey of exploration down that path. What we are doing with pacemakers, deep brain stimulators, is just the very beginning of a most interesting road. We're familiar with these experiments where people are trying to pick up either directly or indirectly brain signals to bypass the people who suffer spinal cord injury. We are familiar with the work that people are doing to try to bypass damaged retina and stimulate visual centers directly. Those interfaces, and I think the best interfaces, of course, to focus on, will be the neural system electrical interfaces, are an extremely exciting area for biology. Because not only do they have major impacts for medicine, they have major impacts for one of the things we like to do the best, communicate. Communicate, understand, and interpret our world. And the far extensions of that interface of the world of electricity, communication with our primary nervous system are interesting, somewhat troubling in their overall ultimate potential, but certainly ones that will occupy much of our attention in the years to come. Those of us who are involved in this research can feel the power of what exists today. We know, no matter what field we are in, that these are unprecedented times of opportunity for those of us who wish to push forward stem cell therapy, interfaces, materials. No matter what area, the opportunities that we can create are boundless. But will they be bounded? The problems we will face, greater than any of the hurdles that are inherent in the technologies themselves, come from without, not from within. I am confident, given our limitless inventiveness, and that is one of our fundamental characteristics. We seem to be unlimited in our imaginations and energy to turn new knowledge to useful advance. But we exist in a broader context, the context of our society, which is beginning to push back on many fronts. It is those bounds, whether we talk about the restrictions on stem cell therapy, which can stifle and in fact are now stifling stem cell research worldwide. It's not imaginary, it's here, it's now, it's real, and it can be a criminal offense to do certain experiments. Horrendous and unimaginable a few years ago, but real today. The restrictions, the debates that are going on right now in Congress about how to fund our healthcare systems can produce a confining noose on our technologies and their ultimate use. These are not idle debates that will not affect your work. They will affect your work. They will affect your work in real time, whether you're working in a university, whether you're working at a large company, whether you're working in a government laboratory, whether you're working in a biotech company. These constraints that we were going to now experience from society will be the major boundaries that determine the impact of our work on human health. And finally, there is a debate, and it's a deep debate that goes to the heart of human nature, which is how good is health? Where do we put health? Where do we put longevity? Where do we put equal distribution of health benefits in the overall scale of human priorities? The answers seem to be obvious until you start to debate them. And once you start to debate them, you realize that there are very, very different views, not only in our country, but in countries around the world. And that those boundaries, as much or more than the technical boundaries that we will bump up against, are the major forces which will shape our medical future for the next 25 years. We all know that that future can be bright. We know there will be many successes. And I'll just end by encouraging you not only to go on with your own work, but to pay attention and become active in these greater forces which shape it. Thank you.
Thank you very much. Thank you. Bill brought up a half a dozen things that will be fleshed out in one way or the other by future speakers, and if you've gone down the list and looked at their bios, you'll see that they're going to be touching on a lot of those issues. The last thing is that he said is really the killer app, so to speak, and that is if when you realize that sort of glibly they say that 90% of healthcare costs are keeping spent in the last 6 months of people's lives, the implication of that, or even altering that so it's not 90%, it's 80%, whatever it is you think of what that means for the development of a healthy world.